Hello, and welcome to this presentation exploring microbiology as a subject at Trinity. My name is Dr. Kim Roberts, and I am Assistant Professor of Virology. I'm going to start by talking about microbiology in general and the course at Trinity. Then we'll look in a bit more detail about two topical areas of study and how we teach them. Firstly, I'll discuss how I teach virology, and then I will be joined by my colleague Dr. Sinead Corr, who will tell you about her area of research, the microbiome. So what is microbiology? Well, microbiology is the study of all living organisms that are too small to be visible with the naked eye. This includes bacteria, archaea, viruses, fungi, prions, protozoa, and algae, collectively known as microbes. It can be argued that we live in a microbial world. Microbes do not need us, but we certainly need them. Microbes play a crucial role in our lives. They are essential for nutrient cycling. Some cause food spoilage, whilst others can be used to make food. As we'll hear in a few minutes, microbes are essential for our health, but of course, some microbes make us ill and cause disease. Thanks to their versatility, microbes can be put to work in many ways, making life-saving drugs and vaccines, the manufacture of biofuels, cleaning up pollution, as well as producing and processing food and drink. Would it surprise you to hear that without the activities of microbes, there would be no bread, cheese, beer, or even chocolate? Recent Nobel Prizes celebrate discoveries with microbes. Drs. Harvey Alter, Michael Horton, and Charles Rice were honoured for their discovery of the hepatitis C virus. And Drs. Emmanuel Charpentier and Jennifer Doudna were awarded the Nobel Prize for Chemistry for their development of a method for genome editing using an ancient bacterial immune response. They created a set of molecular tools that has exploded molecular biology. The field of microbiology has a long and distinguished history and an exciting and essential future. One of the fascinating aspects of studying microbiology is the way in which the discipline interacts and overlaps with other subjects. At Trinity, we take a holistic approach, studying all aspects of microbial replication, genetics, cell biology and biochemistry, the stimulation and subversion of immune responses, and of course both the ability of microbes to cause disease, as well as ways in which we use microbes to combat a wide variety of diseases. For example, during the programme I discuss ways in which human viruses can alter the eukaryotic cell cycle and inadvertently contribute to cancer development. But we also discuss how viruses are now being used as a treatment for some cancers. These oncolytic viruses have been engineered to stimulate the immune response and destroy tumour cells. So if you have an interest in several of these fields, microbiology could be a great fit for you. To study microbiology at Trinity, you would select the TR060 course for the Biological and Biomedical Sciences stream. You would study core biology courses in first and second year, and at the end of second year, you would choose which course you wish to pursue in your final 2 years. The courses in years 1 and 2 are designed to give you a broad understanding of biology, from molecules to cells and organisms to ecosystems, and microbiology is taught within the core courses in years 1 and 2. You would also have options to study additional areas that interest you, such as the immune system and genetics, and there are microbiology lectures in these courses too. If at the end of second year you choose the microbiology moderatorship, then the Moyne Institute of Preventive Medicine would be your base. You would take advanced courses learning about how microbes cause disease and how we can utilise microbes in industry. As in first and second year, you would have the scope to tailor your degree to your interests through your choices of open modules and advanced topics. The microbiology department has a broad and diverse faculty. The research areas of our lecturers and professors include bacterial genetics, how microbes cause disease and immune responses to infection, resistance of bacteria to antibiotics and drug discovery of new antimicrobial drugs, the manipulation of fungi for industry, and using microbes to model human disease, as well as respiratory virus infection and prevention, and the human microbiome. The capstone of final year is the research project, where you would choose to work in one of our research labs investigating your own project for 9 weeks. I'm sure one of the things you are considering when choosing your degree is the types of jobs graduates go on to. From microbiology, many of our students walk into jobs in food and drink manufacturing, the pharmaceutical industry, and biotech companies. Several students have gone on to study graduate medicine, and others have gone into hospital diagnostics and forensic science roles. Many continue their education in master's courses and PhD programs. Others go into teaching, publishing, or become investment analysts or bankers. So there are many careers our students choose where they use the scientific knowledge they've developed during their degree. And also there are many careers that value the transferable skills developed during the programme, such as critical thinking, problem solving, and communication skills. You might find it useful to hear what some of our third-year students have to say about microbiology at Trinity. Chloe said, "There's a good balance between clinical microbiology and the molecular aspects of microbiology." Stephanie said, "If you're caught choosing between genetics and immunology, then go for microbiology because it's the best of both." Emma said, "It was my 6th choice, but if I had to choose again, it would be my first." And Claire said, "They know how to throw a good party." So now we're going to look at a couple of topical areas of microbiology that you might be interested in. Firstly, my own area of virology. The SARS coronavirus 2 pandemic has shone a light on how vulnerable we are to emerging viruses and how dangerous our interactions with wild and domesticated animals can be. Viruses infect all life on Earth, and occasionally a virus can jump from one host species into another, and sometimes into people. These zoonotic infections can cause severe disease, but usually the virus is not well adapted to us and doesn't transmit well between people. Unfortunately, sometimes that isn't the case, as we have seen with recent large Ebola virus outbreaks. And as the COVID-19 pandemic shows, when a novel virus emerges that is efficient at transmitting between people, it can have devastating consequences on individuals and society. This is one of the reasons we think it is so important to integrate virology within the biological sciences stream at Trinity, and we teach virology in more detail than many other courses in the country. In first year, we discuss the basics of what viruses are and their genetic and structural diversity, as illustrated in these pictures. Viruses are simply packages of genomic material, either DNA or RNA, or occasionally a mixture of both, wrapped up in a protective protein or protein and fat coat. Viruses aren't alive, and they are dependent on finding a host cell to manipulate in order for virus replication to take place. Viruses are simple, with relatively few genes, but they interact with their hosts in complex ways. In second year, we delve deeper into the diversity of viruses, and I use a few topical viruses, such as influenza, human immunodeficiency virus, HIV, and SARS coronavirus 2, to compare and contrast the different ways viruses infect cells, replicate, cause disease, and transmit between people. Different viruses rely on different transmission methods to be passed from one host to the next. Some are carried by insects, such as mosquitoes. Some are transmitted through saliva or blood. Others, like influenza and SARS coronavirus 2, are transmitted through respiratory droplets. We also explore the tools scientists have developed, such as vaccines and antiviral drugs, to combat and prevent infections. In third year, we explore a wider range of weird and fascinating viruses, as well as a practical course where students learn how to work with mammalian cells, investigate different methods of quantifying viruses, and explore the diagnostic PCR test used to identify viruses, such as influenza infections. In fourth year, we discuss in detail how viruses manipulate cells during virus replication and the strategies cells use to fight back. We discuss how the host immune response fights infection and how viruses block and hide from those host immune responses. So if you're interested in learning more about viruses, then please take a closer look at the microbiology degree at Trinity. I'm now going to hand over to my colleague so that you can hear more about the human biome.
Why study microbiology? My name is Dr. Sinead Corr, and I am a lecturer in the Department of Microbiology. My lectures focus on the human microbiome, an area of study that has exploded over the last 10 years. We live in a microbial world. Microorganisms are everywhere. Understanding the world of microbes is important for understanding many aspects of life on Earth. Microbes are the invisible heroes of our planet, cycling nutrients, generating energy, and producing oxygen through photosynthesis. Everywhere we have looked on our planet, we find microbes, at the bottoms of oceans, in deserts, and even in Antarctic ice. But what if we were to look closer to home? Microbes have evolved to become a critical part of the human body. Our body is like a microbial rainforest. There are thousands of species of bacteria, protozoa, fungi, and viruses living on and in our bodies. This is called our microbiome. We are learning that these microbial passengers contribute to nearly all aspects of human health and well-being. While our gut is home to the largest volume of microbial residents, we also have microbiomes in other body sites, including the oral cavity, skin, and lung. When you look closer at this microbiome, you have to ask, how human are we? The human body contains over 10 times the amount of microbial cells as human cells. When we look at the genetic material in our bodies, 99% of our genes are microbial. In fact, there are more genes in the human microbiome than there are stars in the observable universe. Interestingly, any two people share 99.99% of their human genome, whereas they share only 10% of the microbiome. Research has revealed that there are millions of genes unique to individual microbiome samples, suggesting that the genetic diversity in microbiome from person to person could be enormous. So how would you go about studying these microbes? The field of metagenomics is working to answer many questions about the microbiome. When we study the microbiome, we talk about diversity, meaning that we not only count the number of bacterial species in our microbiome, but also the abundance of each type. We know that diversity is the key to health. The more diverse your microbiome, the healthier the individual. The human microbiome evolves throughout life, beginning at birth, and we see that microbial diversity increases over time, eventually reaching a plateau, and then declining with age. This usually is at a time when many health issues arise. We know that the diversity of the human gut microbiome in particular is impacted by many factors: type of birth, use of medicines and antibiotics, lifestyle choices, dietary choices, and even geographical location. These factors can cause a disturbance in the microbiome and contribute to loss of diversity. This less diverse gut microbiome is associated with development of a range of diseases. We also now understand that there is a connection between the gut and the brain, often referred to as the gut-brain axis, and the state of our gut microbiome is important for regulating this communication. It's been shown that the gut microbiome can impact on behavior and mood, depression, anxiety, and also contribute to neurological disorders. If you choose to study microbiology here at Trinity College, you will learn about many aspects of the human microbiome and its interaction with our bodies, in particular our immune system. We will consider how the microbiome is influenced by and influences human genetics. You will delve into current research which studies changes in the diversity of the microbiome and how it contributes to the development of disease. By studying individual bacterial members of the microbiome, you will learn about the ability of certain bacteria to cause disease, or how beneficial microbes can boost health. Many of the themes mentioned here are under investigation by my own research group, which is located in the Department of Microbiology, housed in the Moyne Institute of Preventative Medicine. If you would like to learn more about the courses we teach through the Microbiology department, I invite you to take a look at our website.